like this approach, and we tried it out on different environments. Here's Lake Manyara in Tanzania, and there's a gradient, strong gradient in vegetation across that. And the vegetation has a strong isotope range from about -36 to -26. And we measure the deuterium on the leaf waxes of those same compounds, and there's quite a range, at about 60 per mil. But when we apply the scale, the shifting scale of fractionation factor based on the 13C content, we reconstruct waters that's really well behaved and very narrow in estimate. And so that's the approach we've tried to use with our old device samples. Here's an example from that land surface that I showed you earlier. We have measured 13C values, -36, -28, and then we apply a fractionation factor scaled based on this, and we reconstruct water values that are very similar to each other in the landscape. And because they're coeval, we would expect that to be the case. In the lake environment, we can also look at algal compounds like C17. Alkane is from algae, and you can reconstruct water composition. That's kind of cool, but there's a caveat here, and Julian Sachs at the University of Washington, Seattle has really done some really nice work mapping out how salinity in the environment will influence fractionation of these lipids by algae when they're producing them, and it's a really interesting effect. And so if you estimate the salinity and then correct your measured values, you can then reconstruct this. The light gray line is the epsilon that we reconstructed from our measured values. So it can make a significant change. And in this particular environment, we have a lot of variability in the lake level, and we inferred a fairly wide range in salinity change as well. So one of the things that we were interested in is, can we correct our lipids from the lake and our lipids from the plants and try and reconstruct changes in aridity in the environment? And this is the figure that shows that the lipid values reconstruct rainwater that looks like this, and the lake water from the algae is reconstructed to look like that. In all cases, the lake water is more evaporative in its signal than the leaf waters, or the leaf carried water signal. And so we're inferring this as a rain signal and this as a lake water signal. You'll notice when we have lighter isotope values in the rain, that in tropical regions like this, that's often associated with higher rainfall, and that we have a 50 per mil variation between these two. And when it's drier, we have a broader separation. So this is suggesting that the lake is kind of more evaporative than the rain during those dry time periods. So we wouldn't expect them to be parallel lines, but they should actually be at slightly different slopes. And this actually ties into some work that we did some years ago, sort of thinking about lake behavior relative to the waxes coming from the vegetation in the catchment. And this is from a study that we published some years ago now, Pradika Polisar and I, based on a bunch of high elevation lakes. And the idea is that you have a meteoric water line, and then you have a lake water line. You guys have seen that before. And that you would have a leaf deuterium line that's down here that you could correct up to some line based on our fractionation factors. And so we expected to see kind of a lake line versus the leaf line, something like that. And what we found instead is that in many cases, for wide ranges in the lake values, changes in oxygen 18, big changes, we see very small changes in the vegetation carried isotope signatures. So the lakes are kind of dramatic, moving in and out of evaporative conditions over time and space, and the vegetation is kind of carrying a simple signal. And so our interpretation of this in the paper is that that plants grow when it's wet. Right? That's the high-tech interpretation. So the plants are capturing the water at the time that it's raining, and the lake is subject to evaporation year-round, and the algae can grow whenever there's sun. So you have variability in the lake that's captured in the oxygen isotopes of the minerals or the lipids of aquatic organisms that's not captured in the rain-dominated signals carried by leaf waxes. And so they're more of a conservative value. And it's because it's flux-weighted. It's weighted towards the time they grow. And so that, this is my last two slides. Is there a question? Yes?
Yeah, in the previous slide, so the pyramid has a wide, nice range.
This one?
Yes.
Yeah, it's a little noisier, and it's a more open landscape, right? And I have forgotten what we are interpreting that range of values to reflect. I don't know if we had an elegant explanation or not. I can't remember. But it's a really dry environment, right? And so the Paramo?
Super wet.
No, it's super wet.
Yeah. Well, Paramo is correct. We have Paramo.
Oh, it's the grass. It's the Puna.
We
Yeah.
have the water from
Yeah. So the Puna is dry, right? I'm getting confused. I'd have to revisit my own paper. So, and actually, Pradika was the expert on that, because he did the sampling. I just ran the lab. But so yeah, there's more texture there, and I don't know if there's more seasonality that is captured in or vegetation variability. But these are actually a set of different lakes. They're not one point necessarily from one. One line doesn't represent one lake, but it represents sort of a range of lakes within that environment. I'll go read that and I'll come talk to you. have to remind myself of my own work. The last thing I want to say is just this. This is in the Dirk Sasha's paper, and people find that paper kind of confusing because there's a lot going on it. There's a lot in it. This is a review that was published now 5 years ago on what we knew at that time about fractionation and how it gets carried into lipids as a function of environment. And these, if you look in that paper, a lot of the data that I showed you in that previous slide and some of our other studies in Africa and so on are captured in this data set. And what it reinforces to me is that there's a pretty reasonable relationship between rainwater deuterium values and alkane deuterium values that we see captured here. At least when you contrast it with what in that paper was the vegetation signals, which are all over the map, right? So this is a wide range of variability by various plant types. But when we look in the sediments, it gets kind of integrated and smoothed and maybe biased by seasonal processes. And so we get actually a cleaner record or relationship in the sedimentary environment than we do when we go around sampling in modern environments. And at the time of this paper, I think there was a real problem in some of these data sets in that geochemists sampled at about 2 meters, right? They just would go and capture some leaves and then go run them in their lab. And so there wasn't the full appreciation of how to sample plant materials in the natural environment that you guys have learned in this course and that we bring to that. And so I think that there's been a lot of work since this paper that's really tried to push this forward. But I have some faith in the alkanes from lake environments. They integrate a catchment and they are biased towards the wet time, and they seem to behave themselves in a useful way. All right, well, I will leave you with an image of the gorge known as Olduvai Gorge. So thank you very much.